Hi everyone, Professor Gustin here, and we are going to talk about control of microbial growth. A lot of what we've done so far this semester kind of rolls into this topic. A lot of what we talked about, the differences between bacteria, viruses, fungi, what are endospores. We talked about osmosis. We talked about proteins denaturing. We've talked about a lot of things that are going to be part of this lecture. Um, so I think, this tends to be a little bit of a challenging lecture because there's a lot of different parts. And one of the keys is to look at each control method and say, what about that is unique? When would I use that? Why is it different from the others? What special things can it do? The other one is if you forget about osmosis or if you forgot what endospores are, go back and review that because it's going to be really important to your understanding of what's going on in this chapter. So on that note, there's a lot of vocabulary, and this is a table right from your textbook, and it has some of the vocabulary words in here. Sterilization, disinfection, and then decontamination, sanitization means the same thing. Antisepsis and degerming means the same thing. So first of all, we start with sterilization, and it's any process that destroys or removes all viable, means living, or living microorganisms, including viruses. So it inactivates viruses. So when we say something is sterile in the microbiology sense, it means it is absolutely free of microorganisms. You cannot be kind of sterile. I've had students in class, somebody will have a plate and they'll ask me something about it. I said, oh, is that plate sterile? And they'll say, I've heard people say, well, sort of. And I'm like, it can't be sort of sterile. It either is no microorganisms on it or there are. Even if there's one bacterial cell or one fungal spore, it's no longer sterile. So sterilization is a really important term in microbiology, a process that destroys or removes all viable microorganisms. And okay, so we'll continue on. We'll use that term a lot. I want you to compare these two terms, disinfection and antisepsis. And you've heard of chemicals or you've heard of products that you have at home that are disinfectants and antiseptics. So the concept of disinfection is any process that destroys vegetative pathogens, any process that destroys the growing cells, but not endospores. And the reason why we emphasize endospores is that you can reduce microbes on a surface and get rid of most of them, except for endospores, but it's still not sterile. You have to get rid of endospores for it to be sterile. So I want to emphasize that when you disinfect something, you're not sterilizing, not necessarily sterilizing. So disinfection is a process that destroys the vegetative pathogens. Remember, pathogens are microorganisms that cause diseases, but it doesn't remove endospores, so it's not sterilizing. It can also remove microbial toxins too, but this is the main one I want you to look at. This process that removes vegetative pathogens. And the other part of that definition is on inanimate objects. Disinfection is a term used to destroy vegetative pathogens or growing microorganisms on inanimate objects. I'm going to skip down here to antisepsis. It's a really similar definition. Antisepsis or degerming is reducing the number of vegetative pathogens, microorganisms, from living tissues. So like human skin. So disinfection is the reduction of growing microorganisms, vegetative pathogens, from inanimate objects. Antisepsis or degerming is reducing the number of vegetative pathogens, growing microorganisms, from living tissue like human skin. Okay, so you can imagine that disinfectants like bleach can be stronger chemicals than antiseptics like alcohol. These antiseptics have to be compatible with living tissue. If we put bleach on our skin, it's a little strong and it can damage your skin pretty easily. So sterilization, disinfection, antisepsis or degerming. And then the last term we have is decontamination or sanitization. You kind of call that like cleaning. And so it's just some technique where you're going to clean the surface of something. It's a term that's used frequently commercially. So decontamination or sanitization is a cleansing technique that mechanically removes microorganisms. So if you look up here, we used bleach, that's a chemical. But with decontamination or sanitization, we're going to mechanically remove microorganisms, and that usually means washing it with soap or detergent. Okay, so decontamination or sanitization is a cleansing technique. It's using soap or a detergent to remove microorganisms. And we use that term typically in, it says restaurants, dairies, breweries. A lot of times it's related to food preparation, decontamination or sanitization. Okay, so why do we wash our dishes at the end, after dinner? Because we're going to decontaminate them or sanitize them. We're going to wash them and mechanically remove the microbes from the surface using soap. So when you think of decontamination, sanitization, think of soaps. Okay. Here's a picture in your book, and this is one that you could go back to help you kind of quiz yourself and regroup your mind. What we're going to talk about in the remainder of the lecture are the physical things we can do to control microorganisms and the chemical agents that are used to control microorganisms. They put another category here. They said mechanical, but frankly, I would put mechanical under physical. Okay. And I actually do. But come back to this, because what you're going to notice is different treatments can sterilize. Some are used for disinfecting. Some are used for antisepsis. So we said sterilizing would be getting rid of endospores, killing all microorganisms. That's going to be something you do on inanimate objects. Disinfection, remember, that's just killing the vegetative pathogens or reducing the vegetative pathogens, not necessarily killing them, but reducing the number of microorganisms on a surface, and that would be an inanimate object also. Antisepsis is going to happen on living tissues, on animate objects. Okay. So there we go. So come back to that. After you do this, go through all this material, maybe you can look at that and get some insight. The next learning outcome says identify the microorganisms that are most resistant and least resistant to control measures. So this is telling you which organisms are most resistant to microbial control. So there's lots of reasons why we want to remove microorganisms from our environment, from our food, from our bodies, from our bathroom, our kitchen. We're always cleaning up and removing microorganisms. You will see from this diagram that prions are the most resistant to microbial control. We talked a little bit about that when we did prions, but they're actually, remember, they're just proteins and they are extremely stable. Most of the treatments that I will tell you that will inactivate bacterial endospores will not kill prions. You might have to do it longer or something at higher concentrations or higher temperature, whatever. But the reality is we almost never have to worry about prions. This is an exceptional case. We don't have to worry about prions in our kitchens or on our bodies or on medical tools or anything like that under normal circumstances. So even though prions are the most resistant to chemical controls, the lectures that I'm going to give you on this, we're going to use the bacterial endospores as our standard. So for sterilization, if the treatment can inactivate bacterial endospores, the treatment can be used to sterilize. So endospores are our toughest thing to inactivate. The next one is something called Mycobacterium. And Mycobacterium is a kind of bacteria. It's the kind that causes tuberculosis. So there are bacteria that are called Mycobacterium tuberculosis. And they actually have like a waxy wall. So one of the reasons why, especially with nurses, you have to get TB tests and you always want to know if a patient has tuberculosis, because if you cough out these bacteria, they actually can land on a surface and stay there for weeks. And potentially spread to other people because they're so resistant to microbial control. So if you inactivate endospores, you can sterilize. The next most difficult thing to inactivate are mycobacterium. Most people around the house would never need to worry about inactivating endospores or mycobacterium. So in house cleaning kind of situations, we're not typically worrying about sterilizing things. Where we get to, even at the home, is, oh, well, Staphylococcus and Pseudomonas, these are other kinds of bacteria that are common in our environment, and they're a little more resistant than other bacteria. Look down here. Here's the rest of the gram-negative bacteria and the gram-positive bacteria. Gram-positive are a little more sensitive to control than gram-negatives. Remember, gram-negatives have that outer membrane. It makes them a little bit tougher. Remember, some protozoans can make cysts, and they're not quite as tough as endospores, but they're a little bit tougher than some bacteria. And we get way down here where viruses are a little bit more sensitive to microbial control, and enveloped viruses are more sensitive to microbial control than non-enveloped viruses. And they put a note here, SARS-CoV-2 is in this group. I was glad to learn that SARS-CoV-2 was an enveloped virus because I happen to know that enveloped viruses are a little bit easier to kill than non-enveloped viruses. So the bottom line, what you want to know is that different microorganisms have different sensitivities to these environments. If you can inactivate endospores, you can sterilize, and Mycobacterium tuberculosis is also particularly resistant to microbial control. You do not have to memorize anything from this table. It's in your book. I think it just gives you some good, illustrates some of the points I was trying to make. It's talking about if you have bacterial endospores or if you have just vegetative cells, which are the actively growing cells. So here I have Bacillus anthracis endospores and Bacillus anthracis cells, just the bacteria growing. And what it says is if I treat it with moist heat, like boil it or autoclave, you'll learn about the autoclave, you need it at 120 degrees Celsius to kill endospores. Now you might remember that boiling water is at 100 degrees Celsius. So to destroy endospores, you need water at temperature above boiling. To kill just the growing bacterial cells, the vegetative cells, you need it at 80 degrees Celsius, which is pretty hot, but it's not quite boiling. Remember, our body temperature is 37 degrees Celsius. So endospores are 1.5 times more resistant to heat. You can see it takes a lot more radiation to inactivate endospores. We're going to talk about a gas called ethylene oxide. Endospores are much more resistant to being killed by that. And there's some liquids like glutaraldehyde that can be used. And endospores are much more resistant to the things like that. So this is really important to know if you are trying to sterilize something or if you're just trying to reduce numbers. And I want to get it in your mind that most of the time we're just trying to reduce numbers. It's only in special situations where we actually have to sterilize. A little bit more vocabulary. Compare the action of microbial control agents that end in "side" or "sidal" versus "static." And so if we have a word that ends in "side," it usually means to kill. So suicide, homicide. If the word ends in "static," it refers to stopping growth or preventing growth. So there's a lot of words we can put in there. Bactericidal, a treatment that destroys bacteria. When we say it's a bactericidal treatment, it doesn't necessarily mean it sterilizes. It just means it kills bacteria. Okay, it's a treatment that kills bacteria. Whether it kills all the bacteria or not, would have to be determined. A treatment that's bacteriostatic stops the growth of bacteria, prevents bacterial growth. So, and I'll give you examples as we go through the different treatments. Fungicidal is a treatment that can kill fungi, yeasts and molds, and usually yeast and molds, what we're talking about in the microbial world. Virucidal, a treatment that's known to inactivate viruses. Remember, viruses aren't alive, but we say they're active or inactive. So a treatment that's virucidal is a treatment known to inactivate viruses. And a treatment that's sporicidal is capable of destroying bacterial endospores. So the only one on here that specifically will sterilize that all the time is a sporicidal treatment. A sporicidal treatment is treatment capable of destroying endospores. Therefore, it will sterilize. Germicidal, or you might even hear the term microbicidal. It's kind of more of a general term, and it's just a treatment that kills germs, which is a general term for microorganisms. So germicidal or microbicidal is any treatment that kills microorganisms. So something that's bactericidal, fungicidal, and virucidal are also germicidal and sporicidal. That's germicidal too. So all our cidal words up there, they're all germicidal. Germicidal is just a more general term. I think these are my last pair of words. Sepsis and asepsis. Sepsis refers to dirty. So you have your septic tank where your all your waste from your home ends up in, all your waste from your toilets and all ends up in your septic tank. If somebody has microorganisms growing in their blood and tissues, we say they have sepsis. So sepsis refers to dirty or the presence of microorganisms. You put a in front of a word. In biology, quite often it means not. So asepsis means not dirty. And in microbiology, we use that word any practice that prevents the entry of infectious agents into sterile tissues. And not necessarily sterile tissues, but even on the lab bench. You know, you're streaking out a Petri plate and you only want to introduce one kind of bacteria to it. You're going to use good aseptic technique. You'll learn, and so that means not dirty technique, clean technique. So nurses learn about aseptic technique in clinical settings. A patient has a wound and you have to change the bandages. You don't want to lean over it and cough into it and touch it. That would be bad aseptic technique. In the laboratory, you want to sterilize your inoculating loop before you streak a plate, things like that. You don't want to leave your Petri dishes open to the air any longer than you have to. Closing it, preventing other microbes from getting in would be good aseptic technique. So sepsis and asepsis. And that is the end of that section. We'll start on physical control in the next video.